they want to see the virtual concert at TCO Chambers.
What's this morning?
Sorry?
What are we looking at? Female? Oh, yay! Female reproduction? I know.
If you're a mammal, then we'll cover you today.
Okay.
Yeah, I was reading the textbook and I was like, I do this.
Cool. I'm in training for now, so I'm about to. How exciting! Oh my gosh, I was like away from like friends and everything. So I was like, do you know that you're might be below 40 degrees? Like, and I was like sending them pictures of like the diagrams I was drawing, and they were like, oh, this is like good. I know. Look at my toy cells.
Yeah, I'm taking brain and behavior in the psych department, and we're doing sexual differentiation right now. And so he's like gonadotropin, and everyone's like freaking out. And like, all right, like we've been doing this.
Yeah.
All right, let's get started. So first of all, we tried. Exams did not get finished by today, so but they will be finished by Wednesday. We're almost done.
You're like, no.
Okay. So we covered male reproduction last week, and today we'll cover female reproduction. It's both similar and different. All right. So there's there's 4 stages of pregnancy. Not pregnancy. 4 stages of the reproductive cycle. Okay, the first is what is called proestrus, and this is the period prior to ovulation. And everything's happening then. The second period is estrus. And this is when we have ovulation. Now, for mammals, there are 2 types of estrus. There is monoestrus and polyestrus. So monoestrus is what happens when you only have one period time per year. So once per year. So you have a lot of the carnivores only have one estrus period a year. All the animals that hibernate, they only have one estrus period per year. The rest of us are polyestrus. So we have many. And what is key is that you can get polyestrus, you can get mating immediately or shortly thereafter, estrus failure. So after estrus failure. So if you do not become pregnant when you are polyestrus, you can then start the whole cycle again and get pregnant. If you're monoestrus, you miss it, you're done for the year. You got to wait till the next year. Okay? So 2 types of estrus, and then there are 2 types of ovulation. There are what are called the spontaneous ovulators and the induced ovulators. So a spontaneous ovulator is a species in which ovulation occurs regardless of what's happening in the environment. An induced ovulator is where ovulation occurs coincident or because of coitus, which is the insertion of the penis. Okay? Makes sense? What are humans? Spontaneous. Why would we be spontaneous? Why does it make sense to be either a spontaneous or a induced ovulator? When would you want to be one or the other? Yeah?
Monoestrus would want to be induced because it's only at one period a year. You want to make sure it's happening when reproduction is occurring. When sex is happening.
Okay, so definitely if you were monoestrus, you'd probably want to be an induced ovulator. It turns out not all monoestrus species are, in fact, induced ovulators, but many are. Yeah?
If you'd want to be induced if you're not going to find a mate that often. So I know cats are induced ovulators, and sometimes they don't run into mates, but if they do, they'll be able to actually, have kittens then. For example.
Yeah, well, it turns out that almost all of the cats are induced ovulators. I think lions are the exception. But but almost all the cats are induced ovulators, which means as soon as a male is there and you have mating, you have an ovulation. But why wouldn't all species of mammals be induced ovulators? Why would it ever make sense to ovulate when there's no male around? Yeah?
It might attract males. Like, maybe the process of ovulation secretes hormones and pheromones that attract males.
Yeah, but so do induced ovulators, right? So we're not actually talking about attraction. There's all the different types of attraction are present in both induced and spontaneous ovulators. Yeah?
Is it like a vestigial trait from ancestors who used to like do external fertilization? So, like,
Okay. So, well, first of all, you have to go a long ways back to get to the ancestors of ours, of mammals that were not internal fertilizers, right? But try to answer this question here. Which do you think is the majority of mammalian species? Induced or spontaneous ovulators? Yeah, it's overwhelmingly spontaneous ovulators. And yet, induced ovulation makes so much more sense. If the object here is to fertilize an egg, why release that egg unless you know that the male is right there and that you have sperm to fertilize?
it takes like a lot of energy to like keep and like be able to have like a fertilized uterus or a uterus that's able to hold.
Well, that's possible. I'm not sure that it's going to take a whole lot more than cycling again. Yeah?
Does it have to do with reproductive, like the actual, like once you are pregnant, it takes a long time. So that kind of energy, I guess, may be the reason why you ovulate and you could potentially end up getting pregnant or not getting pregnant. I think it's, I don't know, because the cycling of the hormones then.
Not quite sure. But here's the question. So in terms of energy. So let's say that you want to ovulate on day 1 and the male's not around until day 30. So is it more energy to maintain everything from day 1 to day 30? Or to, as we do in humans, get rid of everything and rebuild it in 30 days? I don't know which is going to take more energy. I really don't know. But it's not obvious to me that it's going to take more energy to maintain it than it would be to get rid of it and rebuild it. So it's probably not an energetic issue. Yeah?
Spontaneous is faster. Like,
you don't have to like go through this, like you're like already ovulating, you don't have to go through the whole hormone cycle in order to actually induce it during coitus. It's just there.
Well, it might be easier, but if you're not pregnant, then you've failed. Right? Yeah.
Is spontaneous easier to regulate?
Probably not. As far as I've read, there's no difference really in the regulation of the process. Yeah?
Could it have to do with when mating occurs, spontaneous versus induced ovulation?
What do you mean?
So like, so if mating is more like season based, then you would expect to have like a more regular pattern of ovulation versus if it can occur anytime. But I don't know how to go on from there. So.
Yeah, I think you're closer. I don't think it has to do with seasonal, because a lot of seasonal species, like sheep, that only breed mate in the fall, or dogs. Well, dogs are not a good example because they mate all the time. But lots of other species that are only seasonal breeders also are spontaneous ovulators. So, but I think you're on the right track. Yeah?
Are like the females kind of like looking forward to mating with different males within like a relatively short amount of time? Like for example, because I know that like some male sperms actually like have mechanisms to compete with other sperms who are also in the same female's body.
Yeah. Oh boy, you're the first person who's ever brought that up in class. That is a fascinating topic, but there is lots and lots of data building that a lot of adaptations that take place are for what is called sperm competition. And sperm are actually competing for the egg inside the reproductive tract. And there's some fascinating stuff about that goes on. Certain bird species, for example, they'll shunt. They have a little part of the oviduct where they can actually shunt the sperm into it. And so the first male who mates, their sperm gets shunted in. The second male comes in, and the female then seems to be able to regulate which sperm will access the eggs. Yeah, talk about crazy. Yeah?
Could it be that it's depending on how many babies arise from it? Because induced, if the animal like cats, they have multiple kittens per litter, but for humans like spontaneous, we only average one, so spontaneous you get more out.
That was the other way around. The induced has the litter, and we didn't. But it turns out it has nothing to do with litter size, because you can have induced and spontaneous. Rats, for example, mice are spontaneous ovulators. They have tons of babies. Yeah.
Does it have to do with parenting? That if a male comes in, if it's spontaneous, then the first time that they have sex, it won't result in babies, maybe? And that they have to stay around for longer?
Yeah, that's a good idea, except that both species where the male has considerable parental care, like humans, are the same. There's no difference with those where males have virtually no parental care. Like I talked to you about the voles. The voles that don't like each other, male has essentially no parental care. Where the vole species where they like to be with each other, the male does a lot of it. They're both spontaneous ovulators. Yeah.
Could it have to be with the number of eggs the female has available?
Nope. Yeah.
Could it have to do with the frequency of mating?
Nope. We're running out of ideas, huh? I think it's much simpler than you guys are thinking. Yeah.
Could it be that induced ovulation was it was a mutation that was more beneficial, since most people are spontaneous ovulators and then induced ovulators?
Yeah, but if it was that beneficial, then why hasn't it swept through the entire mammalian clade? It's a good idea, and it probably was a mutation. We think that the ancestral condition was a spontaneous ovulator. But why would it make sense in some cases to be an induced ovulator, but for most situations being spontaneous. Yeah?
The length of time that the egg can like be maintained inside the uterus?
No. All right, I'll put you guys out of your misery. It has to do with access. So if a male is around, then and around almost all the time, then you'd be a spontaneous ovulator. Female decides when to release, when to ovulate, and there's always a male around whenever she ovulates. But think about something like, not a lion, because we always think, when we talk about cats, we either think about lions or our house cats, right? And those are two bad examples of cats. Most cats are not like lions, and they're not like our house cats. Most cats are like jaguars or cougars or things like that have massive ranges, and they exclude each other. And so the only time they ever meet and tolerate each other is to mate, and that could take place a month from now as you're wandering around your territory. And so when a male is not always around, then you wait and ovulate only when you're induced. Does that make sense to people? If the male's around, let's just spontaneously, just let's get it over with. Do the ovulation. If the male's not around, we need to wait. And how do we make sure the male's there? That's when mating occurs. Yeah.
So is there any drawbacks for a species that, for females that can't easily find males to still have induced ovulation? Because that still sounds more energy efficient.
Yeah, doesn't it? So I don't know what it is. Nobody's been able to figure this out. You know, why? Because it just seems to make more sense to be an induced ovulator. I mean, if the whole object is to become pregnant, then let's make sure that the male's there, always. But that's not the way it seems to work. And I think the answer, people have come to this idea that the answer probably is that for most species, there is always a male there. Okay? All right, so those are the first two. There's 4 stages. 4 stages. So proestrus, then we have estrus, and the next thing we have is diestrus. So this is the period right after estrus and or after ovulation and up to the next beginning of the cycle. So this is when pregnancy does not occur. And then finally, the last stage is menses. And this is it's similar to proestrus actually, except now we get the sloughing of the uterine lining. Okay, so this is when we start the whole process and start the process again. By the way, why do mammal females menstruate? Once again, it makes absolutely no sense to lose all this tissue that you're going to want again in a couple of weeks, especially when you realize that you're losing all of this blood. I mean, think about it. How many times do you hear that women get anemic when they menstruate? If they're starting to lose iron, this does not sound like an advantageous thing for evolution to all of a sudden be anemic for a week. So why in the world do we do it? Yeah?
I read somewhere that because pregnancy is inherently high risk, so if the fetus is non-viable, and it's also not a part of you either, so if there's anything wrong with the fetus, then like you need to shut it down and like drove it out immediately, or else like you get really bad consequences.
Yeah, so that's probably very true, but that's not actually the definition of menses. That would be more of the definition of a stillbirth or a spontaneous abortion, because you actually have a fetus there. So at menses, you don't ever have a fetus at all. No fertilization occurred. Yeah?
Because you have to remove the egg that was released for a new egg and prepare it for the next period that you would end up having.
Yeah, but why do we have to lose the entire uterus to get rid of one cell?
Because it doesn't it, no.
That's not going to implant or do anything unless it gets fertilized. That can just get flushed out. We don't have to lose all of that lining, all that tissue, and all of that blood just to get rid of that one cell.
But they would build up if you don't get rid of them.
Well, it just builds up to the right spot and then just sits there.
Then how would a male penetrate to actually impregnate a female if they have eggs that are dead just sitting there?
Oh, the eggs will get flushed out. Plus, how many eggs do we got to get through? I mean, even a year, that's only 11 or 12. There's millions of sperm.
They have trouble sometimes finding it to begin with.
Oh. That's probably a myth, by the way.
Yeah.
Yeah. Okay. So, it turns out that they now classify sperm in terms of 3 different categories. There's the typical sperm that we talk about, which are what they call the egg getters. And they are specifically there to swim up and find an egg. And then they used to have all of these sperm that were all deformed, and everybody said, well, it must be very difficult to make these, and so they're deformed and it's not very good. Turns out that's probably not. It's probably intentional. That these really bad looking sperm get stuck inside the channels that move up through the cervix and act as blockers so that another male's sperm cannot get up. So it's probably intentional. And then there's a third type of sperm that always were these ones when people were looking in the microscopes, you always see these sperm going backwards, right? And they're saying, oh, they're lost. Well, it turns out that those sperm have extra potent acrosomes, and they now think that those are actually what they call now hunter killers. That their whole purpose is to swim around inside the female's reproductive tract trying to find other male's sperm. And once they find the sperm, they release their acrosomes and kill the egg getters from the other male. So that's what we're talking about. There's a huge amount of sperm battles that actually take place inside the reproductive tract. It is crazy. But this myth that, some of the sperm get lost, it's probably not that. They're probably actually hunter-killers going around trying to find other sperm. Yeah. But that's not endocrinology. Where were we? Why do we menstruate? Why do we do this? Yeah?
Is there like a buildup of the blood that has like hormones that are needed?
Not that we know of. Come on, this is a major part of our physiology. Yeah?
The endometrium can become cancerous like really easily because it's proliferating so often.
Well, that's probably true. So, but if we can keep it at just that level, it would stop proliferating. That might actually be better. Instead of getting rid of it every 28 days and having to regrow it. Yeah?
I'm not sure, but could it be something about like the nutrients that the egg uses to replenish?
It's possible, but we don't know. All right. I'll put you guys out of your misery again. The answer really is we don't have a clue. It's in some ways it's really depressing to me. This really is. It's a major part of mammalian physiology, and we still don't know why we do it. Not all species of mammals do what the majority do. So rabbits, for example, have what's called a covert menses. So they do remove their uterus, but they reabsorb it. They don't lose all that iron. They don't lose all that blood. So why in the world do most other species do it? There are some hypotheses, and one of the most interesting ones was proposed about 25, 30 years ago, suggesting that, what is the uterus there for? Is to promote the growth of a fetus, which means that it is essentially one of the best incubators that's ever been invented. And what loves incubators? Bacteria. And so one of the really neat hypotheses is that, to keep it around, basically what you're doing is making this wonderful bacterial incubator. And so if we want to avoid that, we need to get rid of it if we didn't get pregnant. And it makes sense to then regrow it. And that losing that iron is a worthwhile price to pay in order to make sure that that incubator goes away. Now, it's a hypothesis. There are lots of pieces of evidence suggest that might be a good reason for why female mammals menstruate, and there's all kinds of other evidence to suggest that it's not a supportable hypothesis. But like I said, that's one out there. There's others, but essentially we still don't know, which I find absolutely fascinating. We just don't know about this. Okay. By the way, I keep talking about, a month, 28 days. How long is a human menstrual cycle? Yes. What do people say? What's in the textbooks?
I say 28, but it depends.
28 days, right? Here's a fascinating study. I love this study. Okay, this is 30,655 menstrual cycles from 2,300 women. Now look, here we go. Average of this 30,000 cycles was 29 days. So the mean was equal to 29.1 days. But the standard deviation is equal to about 7.5 days. What does the standard deviation mean? It means 66% of the values are within one standard deviation on either side of that mean. So as we subtract that, that's about 21 and a half days. That's about here. Add 7.5, so it's about 36. That's about here. So only 66% of the menstrual cycles were between 21 and 36 days. Another 33% of them were outside that. So this is only 2,300 women versus 30,000 menstrual cycles. So it's not just that one woman has a 21-day cycle, a second woman has a 36-day cycle. Every cycle is different. And there is no way to predict how long the cycle will be. Yeah.
So there's the belief that women who hang out with each other end up cycling at the same time. Why?
Yeah. You know what that's called, by the way?
The Wellesley effect.
Yeah, the Wellesley effect. You know why it was called the Wellesley effect? Because that's where they discovered it. First described it was at Wellesley. So yes, that happens. That's one of the best pieces of indication that there probably is a human pheromone. But nobody's isolated it as far as I know yet. And so we don't know exactly how that takes place. But they start to cycle, so they start to cycle together. But this is a huge variation. And it does bring up one of the most interesting things that I think about in terms of humans. If you think about what makes us different from other mammals, our big brain is one, but there's other species that actually have pretty big brains too. Dolphins, chimps, gorillas. Not to the same extent, but that's probably our biggest evolutionary advantage. But if you think about anything else that's different about humans, we don't have good claws, we don't have good fur, we don't have, we're not big bones, we don't run particularly fast. Some people have pointed out that we run long, and that there's a whole bunch of ideas that they're suggesting that maybe the evolution of humans was in order to basically be long distance runners and run down prey. That we can run farther and longer than any other species. We could run down a horse. It would just, think about it. People actually run. I can't believe they do this, but people actually run ultra marathons, 100 miles in one go. Crazy. That would kill any other mammal that was running. So that's one thing. But the other really interesting thing about humans is that we are one of the few species where the females hide ovulation. Think about that. The woman often herself does not know when she ovulates. And we certainly don't advertise it to the males, physically. Okay? If you look at our closest relatives, chimps, when they ovulate, the huge amount of blood goes down to their rump, and their rump becomes extremely red and pink, so that you can see it all the way through the forest. They are announcing to every male around, I'm fertile. It's my time. And you think about every single other mammalian species that you can think of, the females are telling the males when it's the best time. And female humans do not do that. Yeah.
There have been studies that have shown there seem to be subconscious cues of that as well, because like men look at pictures of the same woman during ovulation and not during ovulation. They almost always pick the one that's ovulating, even though we can't really discern any type of difference between them.
Yeah. So there are subtle cues, but nobody knows exactly what those are. Yeah, it's fascinating. So how do we all regulate this? All right. So now let's get into the endocrinology. So this is the cycle. We've gone through a lot of this already. So this is mostly just a review here. So we have the hypothalamus, we have what's called the tonic area, and the surge area. And the tonic area stimulates the release of GnRH, which goes down to the pituitary. We get the release of LH, FSH. LH is going to go primarily to the corpus luteum, but it's also going to go to the theca interna. We're going to talk about what those are in a minute. What we haven't done yet is talked about what's going on in the ovary. So from the ovary, though, we're going to get estradiol, which is going to have a negative, it's going to help shut down FSH and LH production through negative feedback, but we also get this positive feedback loop through kisspeptin to get that extra surge of GnRH during ovulation. We're going to talk about the corpus luteum. The corpus luteum is going to release progesterone, and progesterone is going to be a massive inhibition on the tonic area and shut down the tonic area. And then finally, we're going to have also through the ovary inhibin traveling back to shut down FSH. So this is all essentially review, but it's important to remember that the stimulus for this also can be from visual cues. We can also get olfactory cues, which is probably what explains the Wellesley effect. We can get negative cues from the pineal. This is melatonin. And we can get negative cues from stress. We can shut this whole thing down. Okay, so the whole goal here of all of this early endocrinology, especially the proestrus, is to develop an egg and get the egg out. So we have to think about, we talked about this in terms of sperm production. We also have to talk about this a little bit in terms of egg production. So we have the primary follicles that are in the ovary. Now, one of the huge differences between male and female reproduction is that in male reproduction, we go through spermatogonia and the production of spermatogonia. The oogonia, the production going through meiosis and that process, in the female takes place as a fetus. And so all of the follicles contain the mature oocytes already. Okay? So then we have primary follicles that mature to secondary follicles. This is where you start to grow a lot of the cells around the egg. And that is going to, as you will see in a minute, turns out to be critical. And then we get some destruction of some of those follicles so that we continue as we go along to have fewer and fewer follicles going through the process. We then have what are called the antral follicles. This is where we start to get the system in the middle here, the antrum. I'm going to talk about that in a minute. And finally, we get to the point where you get the Graafian follicle with the growing egg. So this is the process of getting to the egg. And if we look at each of these the follicles in a little more detail, so we're going through this production of the eggs, and throughout this process, the two things that we're going to be doing, we're going to be producing a lot of these cells surrounding the egg, and that's what's going to create the follicle. And we're going to create what's this center in here that's full of fluid, called the antral fluid. So here's the oocyte, the egg, growing right here. And then this is the follicle. There's two main types of cells. There's these, the thecal cells, and these are the granulosa. And they are separated by this basement membrane. So the thecal cells on the outside, the granulosa cells on the inside. And then inside the granulosa cells, this is the antrum. And so this is your antral fluid. And so this is the follicle that's maturing. Now, the setting up of these thecal cells and the granulosa cells turns out to be very important, because this is where the estrogen is being produced. And so there's a two-cell, both cells are very important. So the way this works is this is your blood vessel. And so the blood is coming down, we're getting cholesterol being removed from the blood and it's being stored in the thecal cell, getting ready to be converted. And when LH is present, it binds to the LH receptor in the thecal cell, and that stimulates the conversion of cholesterol to androgen. And if you remember, androgen is the precursor for the estrogens. But the androgen then traverses across the basement membrane to the granulosa cell. And the granulosa cell then, through this enzyme, aromatase, converts androgen into estrogen. And that's under the control of FSH. And then the estrogen is then released into the blood system. So two entirely different pathway. One pathway through the thecal cell, and the second pathway through the granulosa cell. Okay? So I just showed this. You probably just read this in your textbook. Design the experiments for me that would prove that this actually is correct. Yeah.
You need LH to turn cholesterol into androgen, so if you knock out LH, then you shouldn't get any conversion of cholesterol into androgen. So if you take out LH, you shouldn't see any androgen production. And if you take out FSH, then you shouldn't see any production or conversion of androgen to estrogen, so you should see no estrogen levels but higher androgen.
Okay, so we can do that. So let's take out LH. If we removed LH, we're not going to get any estrogen whatsoever. We're going to lose all of this, right? Because that's the precursor for the estrogen. If we take out FSH, then we will get a buildup potentially of androgen, but no estrogen. Okay? So that would then prove that LH was important for this process and FSH was important for this process. Doesn't tell us that the two-cell model is important. All of that could have been taking place in one of these cells. How do we show that the two cells are actually important as well? So you're part of the way there. Yeah.
Can you take out the
Okay, so one possibility, we could remove these cells entirely. Maybe put the granulosa cells in a dish. Okay? They're not going to be able to produce estrogen unless we provide the androgen. We'd also need to provide something else, wouldn't we? What else would we need to provide? Well, FSH. Yep. What do people think about that? Would that convince you? Yeah?
I feel like I'm only half convinced because I think that the basal membrane, basement membrane may be critical in order to separate the two different types of hormones to show that fecal cell is specifically only to produce androgen and the other one is specifically only to produce estrogen from androgen. Because it doesn't seem like the LH is getting over that basal membrane.
Ah, maybe that's the problem, yeah?
If the granulosa cell doesn't have any LH receptors, it shouldn't matter if LH can get across the membrane, right?
How do we know?
Aren't those the...
How would we know where the LH receptors are?
ICC.
Let's do ICC, okay? So if we do ICC, what we're going to find is that for LH, that they're only on the thecal cells. That's nice. How do we know that LH is important for the production of estrogen at all? Maybe only FSH is required. And it's all in this cell. Yeah?
The LH from the system, see what happens. We don't have it. Just the cycle.
Okay, so that was proposed earlier, but why, to remove LH, we're going to lose this entire process. So it tells us that the thecal cells might be critical here. So if we do the ICC for the LH receptor, so that's not for LH, but LH receptor, right? If we do ICC for the LH receptor, we're going to find it's only on the thecal cells. If we then remove LH and prevent LH from being in the system, we're going to lose our estrogen. So we've now shown that LH is necessary and it's only working on thecal cells. So how do we know that the thecal cells are not the ones producing estrogen? Yeah? Well, we can do, okay, so we can do ICC for the FSH, and we find that it's in the granulosa cells. Who cares? How does that help us? Yeah?
Can you do in situ hybridization to test if there's like estrogen mRNA in your fecal cells?
Well, we can't do that. That's impossible. Right? Why is it impossible? Because it's a steroid. There is no mRNA.
Or use, what is it? ICC.
Or steroids? Yeah?
Could we use a bioassay for whatever's, so assuming that we don't know what's being produced, we get what that is and then we apply it to another system to see if the known effects of estrogen are to happen?
So what bioassay do you want that would show that this is a two-cell system? I'm not sure how that would help. Yeah.
Could you remove the enzyme aromatase and then see if it's estrogen produced and then do ICC for aromatase?
Okay, so I'm not sure you'd want to remove it, but why not just do ICC for aromatase? That gets, I can't remember who said it over there, that gets to, you know, when you ask, so what? The FSH receptor is here. Now, if we know that the FSH is stimulating aromatase, now there's the plug. Okay? Remember, this aromatase is critical for converting androgen to estrogen. If you did ICC for aromatase, you're only going to find it in the granulosa cell. You will not find it in the thecal cell. So now we've proven that the LH is needed to go over here, and we need LH in order to produce our estrogen, and we know that that's in the thecal cell. And now that we've done ICC for aromatase, we know that it can't be the thecal cell that's actually producing the estrogen. It can only be the granulosa cell that's producing the estrogen, because that's the only one that has the aromatase. People starting to see how we can piece this together. Yeah.
So initially, before the experiment, we knew that aromatase was needed to convert an androgen to estrogen.
Yep.
Oh.
Because that's the synthetic pathway. That's how you get estrogen. People see how this works? So once you start to dissect this, now we can figure out that the thecal cells are producing the androgen, and the androgen must be going across to the granulosa in order to produce the estrogen. Yeah.
So is the basement membrane used to filter out only FSH? Like, I'm just wondering why LH goes only to the thecal cells and FSH goes.
I don't know if that's known. The most important thing here is that these are the only cells that have the receptor.
Oh, receptor.
And these are the only ones that have the FSH receptor.
So it doesn't really matter.
So it doesn't matter.
Okay.
So whether or not LH could go across and stimulate the granulosa cell, it probably could, but there's no receptor there, so it's not going to stimulate. So if we come back to this figure then, what we've shown is that what's really probably important for this basement membrane is the distinguishing between the granulosa cells and the thecal cells. And so what we're getting is the androgen produced here getting transported or diffusing across to the granulosa cells, where we now get the estrogen produced here. Yes?
Why is it important that they're separated? Like, if they were both intermixed, what difference would it make?
I do not know. It's just a way that it works. Okay? So one of the things that you'll see here is this is the growth of these follicles. One of the things that you notice when you see the growth of the follicles is that this area is getting bigger and bigger. So what's happening during the growth of the follicles is you're producing more thecal cells and more granulosa cells. If you have more thecal cells and more granulosa cells, one of the products of this is going to be an increase in estrogen. And that's where you get this long increase in estrogen that takes place over proestrus as that follicle is growing. Okay. So now let's start to put the whole thing together. What we're going to do is we're going to go through the 4 stages. Proestrus, estrus, diestrus, and menses, and we're going to look at what's happening with the hormones at the hypothalamus, at the pituitary, at the ovary, and at the uterus. Okay? So proestrus starts. We're going to get a slow increase of release of GnRH. That's going to travel down to the pituitary, where we're going to see an increase in LH and an increase in FSH. That then travels down to the ovary, where we're going to start to see an increase in estradiol release. And then the estradiol is going to travel to the uterus. And here's the uterus. So this is the follicular phase of the uterus, also proestrus. We often call it follicular phase because the follicle is growing. And the luteal phase is for the name for the corpus luteum, which we're going to talk about in a second. And so this is diestrus. And so the estradiol is impacting on these tissues in the uterus, so you start to get what's called the proliferative phase of the uterine lining. So it is increasing, increasing, increasing. So we're going to get an increase in the lining. Okay, and now we get to estrus. At estrus, we're going to see an increase in kisspeptin, which is going to result in a massive increase in GnRH. Oh, I'm sorry, I missed one. So during proestrus, we're getting an increase in estrogen. We're also getting an increase in inhibin. So this massive increase in GnRH is going to travel down to the pituitary. So we're going to see a huge increase in LH and a more modest increase in FSH release. So we look at what that looks like. This is what we see. So this is your days of the menstrual cycle. This is ovulation right here. And we're going to see this huge increase in LH, which then comes back down, and a more modest increase in the FSH. And even during this period, you'll see the LH starts to gradually increase as the amount of GnRH is increasing, but the FSH starts to is decreasing initially during this cycle. And this decrease here is from the inhibin. Yeah.
So inhibin is only targeting the FSH.
Yep.
Is that so? That's also why there's only small.
Yep.
Okay, but otherwise, the kisspeptin kind of overrides.
The kisspeptin is overriding for the GnRH,
and then,
not the inhibin.
Okay. Yeah.
Right? Okay. So what's happening at the ovary at this point? Now we are getting ovulation. So in ovulation, we're going to get a whole bunch of other things starting to happen. So let me come back to here. So the LH is flowing down to the ovary, and there's this area right near in the basement membrane. Nobody knows where it is, but there's always one of them in every follicle, which is called the stigma. And so this is what's taking place. Your LH impacting the thecal and the granulosa cells in the ovary is going to do essentially three things. The prostaglandin, it's going to stimulate the release of steroids, and the steroids are going to be primarily estradiol, and that is going to increase the amount of collagenase. And collagenase is an enzyme that's going to be degrading collagen. And the collagen is the extracellular matrix that's surrounding the cells of the thecal and the granulosa cells, primarily the thecal cells. And so we're going to be degrading the way in which those cells are connected to each other. At the same time, we have the prostaglandins. Remember, we talked about this briefly. The prostaglandins are a lipid hormone. And the prostaglandins go to the fibroblasts that are surrounding the follicle right at the antrum. And they cause those fibroblasts to increase, and so both increase in mitosis and producing extra collagen. So we're getting more and more collagenase. Collagenase is being stimulated by estradiol and by the prostaglandins. So we're degrading more and more of that extracellular matrix. The LH is also stimulating the release of histamine. And the histamine is going to do what's called vasoconstriction. And the prostaglandins help in this as well. And the vasoconstriction is going to take place right there at the stigma, and so we're going to be depriving, these local blood vessels are going to shrink, and that's going to result in a decrease in oxygen available. And so all of the cells, not only are the cells becoming more isolated because we're degrading all the extracellular matrix, but those cells are now being starved of oxygen. And because of that, they go through apoptosis, programmed cell death. And so they die. So if you come back to this figure, we're seeing the death of all of these cells right here in one little spot in this area of the follicle. And those death is what's going to happen is it's going to be becoming very, very weak. And meanwhile, we've produced an enormous amount of antral fluid. And so the fluid is building up and providing a lot of pressure from inside the follicle. And when these cells die, at a certain point, there is no longer the structural integrity in that area of the wall of this follicle, and that follicle will burst. So right here, it bursts, and you have the egg and some of the granulosa cells. So we're going to continue actually producing some of the estrogen, even as the egg is floating down through the oviduct. But this antral pressure is going to force the egg out. And that's where you get ovulation. Yeah?
So can you explain where the fluid and where the pressure is coming from?
So the fluid is being produced by the granulosa cells. And so it's just the buildup. As it keeps producing more and more fluid, it's just building up the pressure. And the pressure will finally get, and it's being contained because we have all of these cells containing it. But when it's time to ovulate, all those cells right there at that stigma die, producing that weak point.
Also, that's where the fluid kind of enters.
It's not where it enters. The fluid is being produced by these cells that are surrounding it. It's what happens, though, is that when these cells die, that pressure just then blows out and taking the egg with it. Yeah?
How, I don't know if we know, how does only one usually end up going through this versus like a bunch of them at once? Because usually you only release one.
That's species dependent.
Oh, that's species.
Yep. So in humans, it's almost always one.
Yeah.
Not always.
Not always, yeah.
But most of the time, it's one. But for a cat or a dog or a mouse or a rat, then they'll have,
But how is it,
7,
do
8,
you
9.
know how it's determined between
Nobody knows
which,
how that.
'cause usually, I know people are always like, oh, it switches between which ovary ends up being released each month, and
Yeah, let's come back to this figure. So all of these follicles are growing. This one is the one that's destined to become the egg.
yeah.
There is a lot of people studying this and a lot of money from pharmaceutical companies that would be available for figuring out exactly which one of those follicles is in fact going to be the final one. Yep. As of right now, we have absolutely no idea of predicting which one of those. And we don't have any idea, we still don't know why there's some sort of a species dependence on how many. We can short circuit it. So IVF, one of the things about IVF is when you inject all of these hormones to increase the production and the growth of these follicles, you can oftentimes get more than one. I've seen some studies that say sometimes as many as 25 to 30 follicles will be growing all at one time. And then they can go in and harvest them prior to ovulation, so that they can have many eggs that could then be fertilized. That's part of the IVF process. So it's not that humans aren't capable of producing more than one follicle at a time. We just don't. And I don't think anybody understands yet why we don't. Okay? So, if we come back to here, what is the ovary producing? It's increasing the production of prostaglandins, increasing histamine, and those are the primary things that are all of a sudden being secreted by the ovary in order to get ovulation. Now the other thing from here is that we see that once the egg is gone, we still get the proliferation and all of the remnants of both the thecal and the granulosa cells. And so this follicle minus the egg becomes what's known as the corpus luteum. And the corpus luteum becomes essentially a huge factory for the production of estrogen, and now a new hormone that's being produced, progesterone. So you get massive productions of estrogen and progesterone. Okay, so let's come back to here. So the uterus, this whole process by the estrus generally takes between about 12 and 24 hours. So it's very, very short. So even when we look at the uterus lining, this is basically a very short line in the whole process of the cycle. So it generally has, by the time of ovulation, the uterine lining has hit its maximum. But this is so short that there's nothing really happening in the uterus. Okay, so now yes?
So the stigma is what, like in the layer where the granulosa cells are?
No, it's really the thecal cells.
The fecal cells?
Yep.
And then that's like the hole where the egg will eventually be released?
Yep.
Okay.
Okay? So diestrus. Okay, the first thing that happens is we have a massive, so just talking about the corpus luteum, that means that we're going to have a large increase in the amount of estradiol being released and a huge amount of progesterone. I talk about that first because these are what's feeding back up to the hypothalamus, and we're getting a huge decrease and basically shutting off of GnRH. I'm going to shut it off completely. Now, this is then going to result in a decrease in LH and a decrease in FSH. All right? Now, let's start thinking about what's happening then back at the uterus. So during the proestrus, we were getting a huge amount of estradiol coming from the ovary. After ovulation, we now have the corpus luteum, which is why it's called the luteal phase. So now we're getting stimulated by estradiol and progesterone. And at this point, what we get is that the uterus starts producing a couple of things. So one of which is we're starting to produce what's called a uterine milk. And that's primarily under the stimulation of progesterone. And the uterine milk is really important for providing nutrients for the egg that's coming down into the uterus before, whether or not it gets fertilized or not, but before implantation. So once it implants in the uterus, it can get its nutrition directly from the uterus. But until then, it relies upon this nutritive fluid called the uterine milk to get the nutrients it needs. The uterus then also starts to produce prostaglandins. And the prostaglandins are going to travel the corpus luteum, where they are going to destroy it. So as one author I was reading one time says, the uterus essentially provides the seeds for its own destruction. Because as the prostaglandins, as the uterus produces the prostaglandins which destroy the corpus luteum, it's the corpus luteum that was producing the majority of the estrogen and the progesterone. So if pregnancy does not occur, the uterus is going to basically cut off its own supply of estrogen and progesterone. So we come back to this one. So what's happening in the uterus? We're getting the increase of the prostaglandins. So pregnancy did not occur, and so what we get is the decrease, the sloughing of the uterine lining during menses. That is going to release the inhibition, because now we don't have this estradiol and the progesterone anymore. So we are going to start seeing a decrease then in the estradiol and the progesterone. But that's going to then release the negative feedback brake. So we're going to start to see the increase in the GnRH again, and therefore the increase in the LH and FSH as we start the cycle anew. Yeah?
Does the kisspeptin stop working after the estrus phase to allow for...
So it's only you're right, the kisspeptin is there to stimulate that surge center. And then it's done. Any other questions? Okay, so let's take a look at sort of one of the overviews. So here's the menstrual cycle where ovulation occurs. So here's our LH. Increases, 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 and get this massive increase for the surge, comes back down and levels off. So this is our LH. The FSH, we see this decrease from the inhibin, and then we see the spike. That's part of the LH surge, which then stays low for the rest of the time. So this is our FSH. We then have our estradiol, low, 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 but then builds up, builds up, builds up. Then we hit the surge center, so we get this massive increase. And then right after ovulation, we see the starting to shut down LH and FSH because the kisspeptin is no longer functioning. And so we see a decrease right after ovulation of the amount of estradiol being produced. And then all of the estradiol is now being stimulated by the corpus luteum. And the corpus luteum is just working on its own, and so we see the increase again of estradiol. And then finally, we see the progesterone. So the progesterone stays low because there's nothing being produced. It's not being produced in the ovary, and then we get ovulation, and now the corpus luteum starts to produce a massive amount of progesterone. And then it starts to decrease as the corpus luteum is destroyed by the prostaglandins from the uterus. Okay? One other way of just looking at this. So we have the growth of these follicles, we then have ovulation, then we have the corpus luteum, which finally degrades. We have the LH and the FSH that then surge right at ovulation and then come back low. They have the estradiol increasing, then decreases down and comes back up with the corpus luteum and the progesterone. And this is all coincident with the proliferation of the uterine lining. The one other graph in here is actually body temperature. And here, LH turns out to be what's called a pyrogen also. It increases body temperature. So you can see a slight increase of about 0.5 degrees Celsius at the time of ovulation when you get that LH surge. So this has been one of the ways in which people have used for a rhythm method for trying to predict ovulation to prevent pregnancies. The problem with that method is that LH doesn't always increase body temperature, and certain other kinds of environmental things can interfere with the signal. So just a funny story for you. When I was in college taking endocrinology, we did an experiment in which the professor gave us all a thermometer to take home. And as soon as we woke up every morning, we took our temperature. Now, it was really for the women in the class, but because men don't ovulate, right? But he wanted to make sure that everybody had the same burden. And then we got all the data together, and guess what happened? The men all ovulated on Saturday, every Saturday. Guess what happened? Yeah?
Over or something.
Wasn't hungover, but you're getting close. Yeah?
Football games?
No, it's body temperature. It was body temperature right as we woke up.
Oh.
Yeah.
For the weekend?
Yeah, it was a weekend, right? So we all were getting up at 7:30, 8 in the morning during the week in order to get to class. Weekend came, slept in. So we didn't get up until 11. Well, that's an extra 3 or 4 hours, and your body temperature hits the circadian rhythm, right? Circadian rhythm pumped up our body temperature, and so you saw a low, low, low for 5 days, and then this spike on the weekend. But it had nothing to do with this process, obviously. It had to do with circadian rhythms. But the same thing happened to the women, too, because they had the same thing. So we never actually saw an ovulatory signal because it was swamped out from the differences in when we were waking up for the weekend. I'm sure you guys don't sleep in on weekends. This probably worked much better for you guys, right? So, this is a danger signal. Don't rely upon this. All right. Oh my goodness, I have 3 minutes left. Yeah, let's start. Okay. Everything that I just talked about is what happens when pregnancy does not occur. So what happens when pregnancy does occur? And here's one of the big things. In fact, I'm not even going to put this up yet. I'm just going to ask you this question. Okay? We have got to have estradiol and we've got to have progesterone in order to maintain the pregnancy. Okay? If you don't maintain your high levels of estradiol and progesterone, you're not going to be able to sustain the pregnancy. You're going to get a spontaneous abortion. Okay. So, how do we keep our estradiol and our progesterone high when we have massive negative feedback shutting down our GnRH? Yep. So we have to keep the corpus luteum. The corpus luteum actually starts to degrade as well. Okay? Yeah. I'm sorry, what? Well, it does a little bit, but we're a long ways from that embryo having an ovary and producing, and the thecal cells and the granulosa cells produce the estrogen. We're at least a couple of months. So the fetus does start to do its own, but not right away. And we need our estradiol and progesterone right away. What has to happen? What do we have to have? Yeah?
Wouldn't you have to inhibit the prostaglandin so then you don't end up having the slipping of the lining? Do you have to maintain that?
Well, we're going to maintain it because we have estradiol and progesterone.
Yeah.
But how do we keep production of that? Yeah?
Maybe there's another thing that increases GnRH and sort of cancels out that negative feedback.
Ooh, almost. Almost. It's not something that mimics GnRH. Yeah?
Does it mimic a kisspeptin?
No. Close though. It mimics LH. Okay? So what we see is this massive increase in this hormone right here. And this is chorionic gonadotropin. And if it's from human, we'll get a little H, so HCG. People heard of HCG before? Yeah, HCG, because it increases right